from us. The next speaker is from the female side, Dr. Deema Hussein. Deema is a well-known scientist at King Fahd Medical Research Center. She is deeply engaged in brain tumors, and we are delighted to have her with us to give a talk. Her title is A Unique Integrated System to Discern the Pathogenesis of Central Nervous System Tumors. Dr. Deema, please.
First of all, I'd like to thank the organizers for inviting me to present the work that we've been doing. And particularly, I'd like to thank the CIPN Center for funding this work primarily. So we are basically a group of committed scientists and neuropathologists and neurosurgeons who are committed to come up with some cures for this disease. And this is an outline for my talk. First, I'll give you a brief introduction on CNS tumors, and then I'll describe some characteristics of the bank that we've been building up, and I will describe a completed study. So, improving CNS tumors prognosis is really a global challenge. Brain tumors are essentially worldwide killers, with ranking 8th between all tumors. In USA, the average percent surviving is about 33%. There's about 120 different types of CNS tumors, and they occur in different locations of the brain, and they are differently pathologically classified. The most prominent is meningioma, which is a tumor in the meninges, followed by other tumors such as glioblastoma. CNS tumors in KSA are on the rise, and survival from data coming up from our hospital here shows relatively poor survival rates. So what's available at the moment to treat such disease? Well, first line treatment is surgery, and it involves, if possible, complete excision. If not, for inoperable, recurrent and aggressive diseases, there are radiation, chemotherapy, and hormonal therapy. And the chemotherapy includes cisplatin and etoposide. The problem is the deficits that you get from both surgery and chemotherapy, so we definitely need to improve treatment, and we need to come up with novel therapies that will target this dreadful disease. So here's a list of the latest outcomes of targeted therapies, which is the post goal at the moment for pharmaco-oncologists, to try to target particular tumors where you have particular mutations. And unfortunately, at best, for adult CNS tumors, you get 66% progression-free survival for 6 months. So the picture's not looking particularly good. In fact, I think the question that keeps hovering in my mind is, are we keep shooting in the dark? And can we come up with, what are the problems that are causing such low survival? So the first problem is that cancer itself is complex, and there are several cancer signaling pathways that interconnect, and they are redundant. The other problem is that most of the genomic information that we've got on tumors is designed on both tumors analysis, so it is slightly missing some of the picture, because CNS tumors are basically heterogeneous. And here's a work that was done last year, and it shows that if you isolate brain tumors and dissociate them into single cells and do analysis, you'll find that here, for example, there's 5 different patients, each with one color. That if you take 100 cells or so from each patient and look at the gene expression profiles, some of these cells will be individualized in their gene expression to the patient, while some of them will overlap with other patients. And on the whole, they are quite divergent between different patients. And if you take 10 cells from one patient and look at the major tumor-associated genes, mutations, you'll find that there is variation between different cells with different mutations coming up in different cells. And this heterogeneity is associated with poor prognosis and poor survival. The other problem is that not all tumour cells are equal and they're not all resilient, and they compete between each other to form different clones. And the last problem is the fact that the way we are testing our drugs tend to be on cell lines. So the way the assessment of newly generated drugs begins with the use of preclinical, I don't know if you can see this very well, but yeah, you can see it. So the starting point is a cell line that comes from a patient. But unfortunately, most of the testing is being done on commercial cell lines. And commercial cell lines, you can see here, for example, 2 primary cell lines, cytogenetic data compared to commercial cell lines. You can see a near-diploid kind of features for primary cell lines that have just been initiated from patients compared to commercial cell lines where you can see almost every cell is representing its own. Also, commercial cell lines, they don't do very well in aggressive assays and they don't grow very well in animals without help. So how can we overcome this problem? And I think, from a pharmacology perspective, we need an integrated approach where we have personalized and representative models, where we can look at functional genomic profiles and functional mutations, where we can understand gene expression diversities between cells and between patients. And then we can test for effective therapies and resolve resistance, hopefully. And the first step to such a process is accumulating samples. So in KU, we managed to collect samples where we can divide the samples into 4 different types. Tissues for RNA and DNA analysis, blood samples for DNA analysis, tissues for in situ analysis, and generate cell lines where we can potentially test the drugs. This is the characteristics of the tumor bank so far. We've got a range of tumours from meningioma to the high-grade astrocytic tumours, you can see here. And the age goes from pediatric to very old, and, to over 70s. And the majority of our patients are females at the moment. So the story with the cell lines is that in 2 years we've attempted 90 different tumours, and technically we managed to retrieve 81 cell lines, but 40 were challenged in cell culture and 41 survived. And these are basically the types of cell lines that we managed to retrieve. Now I'd like to talk about an example of how we can integrate the data from these different samples to actually come up with some meaningful results. So the work we started with is meningioma, and we looked at 17 different types of meningioma. So meningioma can occur in different locations of the meninges, and pathologically there's 15 different variants, and they range from grade 1 to grade 3. And these are clinical profiles of the patients from which the cell lines and the tumor samples were retrieved from. Dr. Fahd Al-Hamdi, a neuropathologist in the hospital, he classified all these tumors and confirmed their characteristics. And so the first point that I started with is looking at the cell culture itself. And one of the basic tools you can look at is cell shapes, because cell shapes represent a repository of information flow of cell signaling system in the cell, and it's often related to its function. And of course, although in normal cells you can see particular definite shapes that are related to functions, you will not necessarily see that with tumor cells. But we managed to get some kind of index where we can see frequent types of shapes of cells in all of the meningiomas that we studied. And we call this index from A to F according to the type. We went on to check whether we looked at the percentage of these morphologies across the first 4 weeks of adaptation of the cell culture from these patients. And we find that adaptation occurs within 3 to 4 weeks, and that regardless of the media that you use or the concentration of growth FBS or glucose, you tend to get sort of a standard morphology in terms of the percentage. And when we looked at all the different types, all the different patient cell lines, we found that they tend to fall into either a monomorphic type with a monomorphic type with a particular D morphology, and they look, if you like, this sort of morphologies, and a pleomorphic type where you can see both spindle, bipolar, and the large cytoplasmic cells. And the first question I asked, does morphology affect drug resistance? And as I showed you earlier, cisplatin or etoposide are constantly or can be used for treatment of CNS tumors. And the first answer is yes. There is a difference between their response to these drugs. And here you can see a graph representing 8 cell lines for each group, where you can see that the D type basically survive less than the BD type in both cisplatin and etoposide. And first, I wanted to ask, why might that be? The most obvious thing is that they don't die well. So I stained cells with nuclear caspase 3, which is an apoptotic marker, and you can see that BD cells have less expression of caspase 3, and nuclear caspase 3 is less frequent in BD cell lines. What else could be going wrong? Well, we've known literature for a long time now that cancer stem cells are resistant to drug treatment, and there have been studies that show that they have been resistant to cisplatin and etoposide. And there are markers that you can use for such cells, such as CD123, SOX2. And this is why I went on to look at these markers in our cell lines, and here's an example of 4 cell lines, 2 from each group. And you can see the staining of CD123 SOX2, nesting Ki67, the minter and frizzled line. And we have actually seen those cells in all of the cell lines. But then when we went on to quantitate, we found a difference in SOX2 CD123 cells and Ki67, suggesting that BD cell lines have more cancer stem cells. And these cells survive with prolonged drug treatment, and you can see here the survival of these cells even after 12 days. So where do we go from here? We thought we wanted to integrate this information. We went on from the cell lines of observation here, and we went to look at the tissue. I remember we collected this right from the beginning alongside the cell line initiation. And with the help of my colleague, we managed to analyze data for six RNA expression. And here you can see that gene expression hierarchical clustering grouped the tumors that had generated BD cell lines separately to those that generated D cell lines. And that D cell lines are closer to normality than BD. And when we looked at differential pathways between the 2 types, we found that top pathways were stem cell related and top genes were stem cell associated. But I wanted to particularly go through one of the AGR2, which we found was highly expressed in BD cell lines, the ones that are resistant. And AGR2 is an important protein in protein folding. It's upregulated in several tumors, and it's been shown to suppress p53 activity as well as being associated with metastasis. So the idea was to go back from the tissue RNA information and look in our tissues and corresponding cell lines. And this is what we found. The AGR2 is upregulated nicely with BMI, which is a stem cell marker. And we picked 5 different areas within the tissue of one of the cell lines, or one of the tissues. And you can see that there is a variation between the tissues as to how they are being expressed, but relatively, it is high expression. When we went back to the cell line, it's the same story, where you can see AGR2, BMI, and they are colocalizing, and almost 65% of the cells in culture are expressing both. So in conclusion, we managed to generate a tumor bank and established 41 primary potentially proliferating cells. A morphology index was generated, which could also be useful for other types of tumors. Morphological patterns of cells can be used to monitor early adaptation, which is important because it's a method that uses less number of cells, which means we can grow our cells and monitor them at the same time, which is vital for the beginning of culturing. BD cell lines, meningioma cell lines subgrouped into monomorphic type or polymorphic type. BD cell line is tolerated cisplatin etoposide treatment better than D cell lines, and they are associated with poor initiation of apoptosis. The cancer stem cell population is more frequent in BD meningioma cell lines, and they survive cisplatin etoposide treatment. And also, the differential trend seen in the cell lines is mirrored in the gene expression profiles of the tumor tissue samples. And so you can see that through an integrated bioanalysis of meningioma tissues and cell lines, we could detect novel genes that might prove valuable for classifying variants of meningioma predicting aggressiveness. Finally, I'd like to thank all the people that helped in this work, and thank you for your attention and listening.